So this is the third session for this assignment solving class. In this week you have studied about enzymes, enzyme kinetics and other microbial reactions. So today we are going to solve the question related to these topics only. Let's come to the first question. My screen is visible?
Yes, ma'am.
Yes, ma'am.
Okay, perfect. This is the first question. Which of the following immobilization technique is a low cost method and has a wide range of application? So they are asking two things. Which one is a low cost method and has a wide range of application? Means you can use that in many applications. So what are the options? First one is adsorption, second covalent binding, third entrapment, and all of the above. So this is very simple question because they are focusing on low cost method. So from all these three techniques, adsorption is the most cost efficient technique and has a wide range of application, not in enzymology, even for adsorption of any other general molecules also we use this method. So for this question answer is A. Now come to the second question. Hopefully you have seen these images in your video lectures. So we have to identify which type of immobilization technique is given in figure A and figure B. Let's read the options. First one, from this figure A, what you have, it is showing like that these patterns are there, and in this sheet pattern enzymes are entrapped or immobilized in this. So this type of technique or method of immobilization is called matrix entrapment method, because this is the matrix having different sizes or maybe similar sizes of patterns in which enzyme comes and sit in these places, means it get entrapped in the provided space of this size. So this figure A is representing matrix entrapment, and then figure B. In this figure you have observed that they have given that this is a semi-permeable membrane. So this is only a one boundary and all the other enzymes, all the enzymes are entrapped or immobilized in this space only. Means this is an example or this is a type of microencapsulation. So for this question answer is B. First one is showing the matrix entrapment and the second one is showing microencapsulation. Okay, so answer to this question is B. Everyone please try to join on time. Now let's come to the third question. What is the correct sequence for the part taken by substrate traveling from a liquid to the reaction site in an immobilized enzyme? So what they are asking for the whole pathway that a substrate take when it move from a liquid to the reaction site in an immobilized enzyme. Means enzyme is immobilized, maybe in matrix form or in capsule form, but it is entrapped. So let's read the options, then we have to arrange them in a sequence, means which process is occurring first and after that which one is occurring. So let's read option A. Diffusion from the surface of the particle to the active site of the enzyme in an inner support. Means what they are saying that now let's read the second one. transfer from the bulk liquid to a relatively unmixed liquid layer surrounding the immobilized enzyme. And third option is diffusion through the relatively unmixed liquid layer. So it means this is a liquid in which you have substrate and then you have enzyme. So what is happening from initially, what happened, it will try to move from the unmixed liquid layer to the surrounding of immobilized enzyme, because first initially what it did, it tried to go nearby surface to the enzyme so that it will go and bind to the enzyme. So for binding to the enzyme, first it have to go there to the nearby surface. It means that the transfer from the bulk liquid, the solution that we have, this is we are calling it as bulk liquid, to a relatively unmixed liquid layer surrounding the immobilized enzyme. So like this is the enzyme. So side the enzyme there is a layering of the unmixed liquid layer. So first what happened, it will try to move from the unmixed layer of the bulk liquid to the nearby layer of the enzyme. Then what will happen, diffusion, because now we know that this is an unmixed layer, so it will try to move from this unmixed layer to the enzyme. It means what is happening here, diffusion. So diffusion occur through the relatively unmixed liquid layer. Then we'll read the third option, diffusion from the surface of the particle to the active site of the enzyme in an inner support. So finally it will go to the active site of the enzyme in an inner support, means in which enzyme is trapped. This can't happen like it will directly go to the enzyme. First it have to go to the nearby layer of this enzyme, and then it has to finally go to the active site of the enzyme. So this is the sequence they are asking. So what is happening first, initially this transfer from bulk liquid to a relatively unmixed liquid layer surrounding the immobilized enzyme. Then what is happening, diffusion through the unmixed liquid layer. So this process is occurring at second, and finally the active site, it is reaching to the active site of the enzyme. So this one is the third part. Now arrange them according to that. B, C, A. So option A is correct for this question. So correct answer to this is A. Now let's come to the next question. The linearized form of MM means Michaelis-Menten equation can be represented by Lineweaver-Burke equation. Which of the following is the expression for Lineweaver-Burke equation? All of you have studied this Michaelis-Menten equation, and hopefully you also have studied this Lineweaver-Burke equation. So first we must know what is the Michaelis-Menten equation. It is mainly represented as V equal to Vmax into S upon Km plus S. So what they are asking about this Lineweaver-Burke equation, what it is when we represent it in a straight line, means reciprocal of this Michael-Mentus equation. So what we have to do, we have to reciprocate this equation, means we have to invert this, like this V become 1 by V and this become Km plus S upon Vmax into S. 1 by V. Now solve this, put the denominator separately, like Km upon Vmax into S plus S upon Vmax into S. So S will cancel out. This will become our equation, that 1 by V equal to Km upon Vmax into this S is with Vmax plus 1 by Vmax. This is the equation that finally come for Lineweaver-Burke equation. So let's check with the options. Option A is 1 by V equal to Km upon Vmax into 1 by S. Here this is 1. Plus 1 by Vmax. So option A is correct. But why we need to do this and why it is important? This is really important because when you study about the different type of enzyme inhibition, or when you want to represent the Michaelis-Menten equation simply in the linear form, we require this, and we compare this with y equal to, you all must know about the graphs of this equation also. We compare this equation with this form, y equal to mx plus c. So in this, how we plot, we consider y equal to 1 by v, and okay, this is 1 by s, this 1 by s is our x, and this km by vmax, it becomes slope, and this 1 by vmax equal to intercept. So you have to plot the equation between these two parameters. So you all must know about the graphs also, because it is really important to solve the questions related to enzyme kinetics, enzyme inhibitions, and other questions related to enzymes. Now coming to this, yeah, answer for this question is A. Now let's come to the next question. Which among the following statement is not true for the solid matrix for an immobilized enzyme? My voice is clear now? Or if you feel any problem, you can ask me in between also. Okay, let's read the question again. Which among the following statement is not true? So this you have to focus. They are asking not true for the solid matrix for an immobilized enzyme. What they are asking, that you have a matrix in which you want to entrap or immobilize the enzyme. Which of the properties not required? So first one is, it should be physically strong and stable. So if it is stable, it's good. It should be cheaper. Everyone wants to make the things cost effective, so if it's cheaper, it's really good. Someone answered, just a minute, be Nitin Kashyap. So you are answering for which question? Okay, let's read the next option. Am I audible?
Yes, ma'am.
Yes,
Okay,
ma'am.
Yes, ma'am.
let's read the third option. It should not be inert. They are saying that it should not be inert. It means if it's not inert, then it may happen that it will directly or it will react with the enzyme. So this is not required. Our matrix should be inert. If it's not is there, then this option is right. Like it, this is a property required, but they are asking for not true. So answer to this question is C, because that should be inert so that it won't react with the enzyme. So this is not the property that is required for the solid matrix. Now coming to the sixth question. Usually we solve around 10 questions every day. and you have any queries regarding these questions or any other questions, then you can ask me. Now let's read this question. If the Michaelis, this is representing Michaelis-Menten equation, okay, is described by V equal to V max into S upon Km plus S, then under what condition will V equal to V max? So hopefully you have studied all the three conditions when V equal to V max or when V is not equal to V max. So you must know all the conditions when these type of things is possible or when it is not visible. So let's solve. Suppose this is V equal to V max into S upon Km plus S. What they are asking? They are asking that it will become P equal to V max. So it means that this part should become 1. And how it will become 1? For this, Km plus S need to be 1. Means S is equal to Km plus S. This can be satisfied when value of Km is almost 0, but it's not exactly 0, or you can say this is very small when compared to this. So that addition to this S, addition of this value of to the S will not affect the value. It means like suppose S is equal to 1 lakh or 1 million and Km is equal to around 0 point something, 0.5, or it's 1. So it will not majorly affect the value of S. So for this equation to be satisfied or this condition to be satisfied, what should require? That Km should be very small as compared to S, so that it won't affect this value and it will become 1, and when it will become 1, V equal to V max. Okay, so answer to this question is that Km is very less than S, that is B. Similarly, you should also know about the another condition, that what will happen when S is very less than Km. Then what will happen? This equation will become V equal to V max into S upon Km. This S will become negligible almost when S is this very less than Km, so it will become Km. So this will become the final equation when the reverse condition will occur. But in this question they are asking about V equal to V max. So for this, what it is required? Km should be very less than S. Okay, it's clear. Okay, now coming to the next question. For this question you required pen and paper. Let's read it. For an enzyme catalyzed reaction at room temperature, the maximum velocity of the reaction is this: 1.4 into 10 raised to the power minus 3 molar per second. And the velocity of the reaction is this. If the initial substrate concentration is 0.44 molar, what is the rate constant? So first we must know what is given and what is required. Let's write what they have given to us. First one is maximum velocity. It means V max they have given. So that is equal to 1.4 into 10 raised to the power minus 3 molar into second inverse. Then what the next thing they have given? The velocity of the reaction means V. This is not the maximum velocity. So this is 1.25 into 10 raised to the power minus 3 molar into second inverse. So let's read further. If the initial substrate concentration means S, or you can represent it as not, but because They are not talking about the reaction change or how much it will change, so we'll consider it S only. So if the initial substrate concentration is 0.44 molar, what they are asking, rate constant means K. So simply we will apply here Michaelis-Menten equation. You have to solve this equation, you will get the value of Vm. Now let's solve it. This is 1.25 into 10 raised to the power minus 3. Because the units here are same and the substrate concentration is also in molar, we don't need to change it. But you have to take care of this really. This is really important. If the units are not same or they are given in different terms, then you have to make all the units of the given components in a similar type. Like if they have given molar, so you should all the reading, all the these should be in molar only. So this is V. Let's put the value of 1 into SS 0.44 K plus SS 0.44. So these units are really important. Now solve this. K plus 0.44, it will become 1.4 into 0.44 divided by 1.25. So now anyone tell me the value of this, how much it will come? Everyone please try to solve these questions with me or try to solve it by yourself, because then only you will get to know and know how to solve these type of questions. Now please tell me the value of this. This is 1.4 into 0.44 by 1.25. Sorry, hard is only 0.5. So which value you are telling? This one. This is 0.5, or you are telling the answer to this question?
After multiplying 1.4 into 0.44 divided by 1.25, we will be getting this 0.4928, ma'am.
Okay, actually your voice is very low, so I'm not able to understand that clearly. Okay, I'll write it. This value of K finally it will come around 0.054 molar, because here substrate concentration is in molar, so it will also come, yeah, it will also come in molar. 0.052, yeah, it's fine, because it will round up. In the options if you rate, there is only one option, that is 0.053. So answer to this question is A. Everyone understood this question? Okay, this is really simple question, but they will, what they will do, they will change the condition. They will give you in the, they will not give you the exact value, they give you in the terms of V and other ratios that substrate become half or double. So these type of question you have to practice. Now let's come to the next question. So what they have given, that given an enzyme with a Km is equal to 10 millimolar and Vmax equal to 100 millimole per minute. and S is given 100 millimolar, that is substrate concentration, how will the ratio of V change if V max is double and Km is reduced to half? So they are asking how it will change when you double the V max and Km is reduced to half. So it means V max will become, in second case, this V max will become double of V, that is 200, and that Km become half of Km, will become, that is 1 by 2 into 10, that is 5. So let write it as V1 and V2. So V1 equal to V max into S upon Km plus S. So this V max, what they have given is 100 into S. Although you don't need to solve it properly, you only need to know in the ratios. So what is Km? Km is 10 plus S. What is S given here? It is 100. This is for V1. And when you find the V2, this is V max. V max become double, that is 200 into S. They have not talked about S here in the second case. So what you will do? You will consider it same as you considered in the first case. Km has become half, that is, you can write it 5 plus S, that is 100. So when you divide it, V1 by V2, it will become, if you consider this 105 or 1010 almost similar, not exactly but almost, then it will become like 1 by 2 of V2 V1. It means, and 1 by 2 is round of around 0.5. So what is will become V1 equal to 0.5 into V2. So this is the right answer for this question. You don't need to calculate this properly because you only want to know the ratio, how it will become. It will, because when it is doubled or half, it will become, hopefully it will come in the multiples of 2 or 1 by 2. So answer to this question is V1 equal to 0.5 of V2. But you have to you have to take care of two things in this. First one, that you should not confuse between V1 and V2, because sometimes if you change this value, then the answer will change. It will become twice of. So you have to check these things with the options also and with the questions also. So answer to this question become
Excuse
B.
me.
Yeah, that's B. So this is the ninth question. Yeah. Let's read the question. The growth of a microorganism on glucose is described by the monod model, sorry, in which they have given mu max equal to 0.8 per hour and Ks is equal to 0.1. If the initial biomass concentration in the batch fermenter is 0.8 gram per liter, so this is really important that they have given initial biomass concentration which is 0.2, then the minimum time required to quadruple the biomass concentration would be. It means that biomass concentration become 4 times. So if initial concentration you will consider is x, then it will become 4x, and they are asking the time taken for this. And you know that monod model, we usually consider it similar to Michaelis Menten equation. But in this equation, we'll consider the very basic formula for this batch fermenter, in which mu, that is rate change, equal to 1 by x of dx by dt. Because how we defined that it is a rate equal to rate of change of biomass per unit time. So when you this is the very initial, that from where we start calculating the various formulas and answer. So we are starting from here only. You can directly use the formula also, but let's try to solve from here only. That mu into dt equal to 1 by x of dx. So both the side differentiation is given, so we'll take the integral to remove that. So it will become mu into t. This integration of this 1 by x is equal to log of x. We can write ln x. Here we also write the limit of from x naught to x, that initial concentration of the biomass is x naught and finally it will become x. Similarly, because here we are integrating with respect to time, so for time we will write from 0, that initially, and it will reach to a particular time t. So it will become ln x minus ln of x naught. So this become mu t equal to x naught. You can directly use this formula if you want. Now it become very simple. Now we only have to put the values, because what we have to find, we have to find the t. This ln, this x to x naught, we also know that initially x naught, finally it will become 4 times of this. And we are considering here at is a that it required as a minimum, so this mu we normally consider equal to mu max, but not in every case, only in special cases we'll consider this. So now time, it will become 1 by mu max, that is 0.8 ln of x, that is 4x naught upon x naught. So when you solve this, it will become ln 4 upon 0.8. Now solve this. Solve this and let me know what it will become. Even you should know that ln 4 can be written as 2 of ln 2, because usually we don't remember the value of ln 4, but ln 2 value everyone remembers. So you can convert in this form. Now finally tell me the value of the time. So someone replied 1.73. Okay. Any other student want to answer this? Yeah, someone also answered D. So everyone is getting 1.73. If someone is getting any other value, then please let me know. Or if you are finding any problem solving this question, please tell me. Okay, so answer to this question will D. Yeah, this is the right answer. Now let's come to the next question. What it is saying, the following equation describe an enzyme inhibition reaction. This you all must know. All you have studied about enzyme inhibition reaction. Okay, we will discuss it. First, we will read the options. So what they are asking, which of the following statement can be rightly deduced from the above equation? From the above equation means this equation. So what they are saying, the above reaction represent an uncompetitive inhibition reaction. So is this the example of uncompetitive? We'll discuss it. In the above equation, I binds to free enzyme and compete with S. Here they have written that I bind to free enzyme only. So this I, is this only binding with E? No, it is also binding with ES. So here it will become wrong and compete with S. Increasing S can overcome inhibition. This can be true, but initially it becomes wrong. So this option B is wrong. Both the statements are correct. That's not possible because second option is wrong. Now we have two options left. First one, that it is a uncompetitive inhibition, and the D option is none of the statements are correct. Just a minute. Everyone please try to answer this question. Which type of inhibition it is? Because all of you have studied this enzyme inhibition. Okay, let's start discussing. We have studied enzyme inhibition. There are three type of enzyme inhibition. Means there is something or this compound. We usually represent it with I because as an inhibitor, from the name itself, we are taking this as. So when it will inhibit the enzyme. Suppose in this given equation it is reacting with E enzyme also, and it is also reacting with ES. So it is inhibiting in both the ways, by reacting with enzyme and by reacting with ES also. So this type of inhibition is called as non-competitive. Okay, when only the enzyme is inhibiting this part, means this inhibitor is only inhibiting the enzyme part, then it will become competitive. Means it is directly competing with the enzyme itself. Now when this, there is third case is also possible, when this I means inhibitor will react with, sorry, will inhibiting or react with ES, this enzyme substrate complex. It is not reacting with enzyme, but it is reacting with this ES only. So this type of inhibition is called as uncompetitive inhibition. So it is clear, these three type of enzyme inhibition we have to study. So this figure is showing which type of inhibition, that is non-competitive. So from this statement A also become wrong. So answer to this question is D. Everyone understood this question? But one more thing we need to take care in this type of equations, or when the enzyme inhibition is occurring, that you should know the curves regarding all three type of this enzyme inhibition, whether it is competitive, non-competitive or uncompetitive, because that is really important and sometimes it directly asked in the questions. Because when this enzyme, suppose this inhibitor is reacting with enzyme, so it will change the rate of this equation. So from due to this velocity or km, these two factors will change because of these type of inhibition. So we must take care which type of inhibition is hampering which parameter. So let's discuss that also. Answer to this question is D, that we have already discussed. Yeah. Lineweaver-Burke plot for all the three type of inhibition. Because this is really important, that's why I am discussing it again. First one is competitive inhibition, means this I is reacting with enzyme it is competing directly with the enzyme. So what will happen when we draw this curve 1 by V and 1 by S for this Michaelis-Menten equation? Suppose this is the curve of a normal, or you can see that blue curve in the given figure is of non-inhibitor, means when there is no inhibitor is used, this is the curve. So where it will cut, it will become Vmax or 1 by Vmax, and similarly here it will become 1 by K. So what is happening here when the inhibitor is used? With the inhibitor, this red line will come, means Vmax, because this is cutting at the same point, this y-axis. This is y-axis and this is x-axis. So Vmax remains unaffected, means it will not change the Vmax of the reaction. Now let's see how it will affect the K or the rate constant. That Km is increased because in the graph it is showing that it is going back, or you can say that here is -2 or here is -3, but this point is not representing directly Km, it is representing 1 by Km. So when you reciprocate this, that is 1 by 2 and 1 by 3, so what will happen? This Km, this value is more, sorry, it is in minus. So when we consider this with minus sign, this value will become more, so it means Km is increased. Okay, so this is the case of competitive inhibitor, when the enzyme is only, sorry, when the inhibitor is directly linking with the substrate by inhibiting the enzyme. Similarly for the uncompetitive reaction, when we find this uncompetitive reaction, it means it is reacting with ES. This reaction of I is occurring with ES. So what will happen in this? Km is reduced and Vmax is reduced. Means this blue line is when there is no inhibitor and red one is when inhibitor, means it will become a parallel line, but in this both the things reduced, Km value also and Vmax. And in the non-competitive or mixed inhibition, we also called it as mixed inhibition, you can see that they are meeting at the same point at the end. It means what it will represent 1 by K max. So it means Km remains same and Vmax, because this point is above this and we have reciprocate of Vmax, so it means Vmax is reduced in the case of inhibition, that is non-competitive inhibition. These two parameters you can learn, but you have to understand how this will changing in all the three type of inhibitions, because this is really important and I want everyone to understand this concept. So if there is any query, then please ask. Okay, let's come to the next question or last question for today. Let's read this question. For the given reaction, the initial concentration of S, means substrate, is given as 200 gram per liter. If 50% of S is converted into P, means substrate to product conversion occurring only 50%, with the help of enzyme E, calculate the volume of the reactor required to produce 15 kg product per day. What they are asking about the volume of the reactor to produce 15 kg product per day when the reaction is operated under CSTR condition, because this is also important. When it is batch or any other type, then the parameters will change. So what they have given more, that the reaction follow Michaelis-Menten equation, that is Vmax equal to 0.5 into 10 raised to the power -2. Similarly Km and Y or yield coefficient is also given. Let's start solving this question. First, we must know what they are asking and what they have given. So they are asking about V, that is volume. Here V is not representing velocity, so you have to take care of this. They are asking about the volume. Normally volume is defined as equal to we usually define, or you can flow rate into time, or flow rate is usually defined that the volume by T. So we require these two things for finding the volume. First one is flow rate, second one is time. So how do we find the F? That is the most important part. And the second most important thing that is given here is CSTR, because we have to take the formulas from the CSTR condition. Let's start this question. For CSTR, when the continuous state tank reaction is occurring, normally what happen? This, or let's write it in the form of T. It will be easier. T equal to S naught minus S upon minus of RS. Okay. And because here it is also given that only 50% of substrate is converted, so that final S, it will become S naught into 1 minus, you can write it X, or because it is given here 50%, so you can directly also write it 0.5 S naught, or you can find this X naught value is equal to 0.5. That is the conversion factor that we consider here. So S is equal to 0.5 of S naught. So now put all this value here and let's find the time. This thing is also required here, that RS. So how RS is defined in CSTR? Anyone know? Or what it is? It is the rate that we usually defined as, I'll write it separately, RS equal to Vmax into S upon K plus S, that we usually taking here. So this time it will become S naught minus S upon Vmax into S divided by, so it will become here Km plus S. So let's solve it here. So when you find out the time using this formula, putting all the values given here, that Vmax is given, Km is given, S is given, and S naught, S we have found in the form of S naught. So putting all these values, this time will come. One more thing you have to take care of this, that here this unit of S is given in gram per liter, but other readings, that Vmax or Y or Km, it is given in kilogram per meter cube. So this gram per liter also need to be converted into kilogram per meter cube. But what this conversion is really easy or equal, because gram per liter is can be equated equal to kilogram per meter cube, because when you convert gram into kilogram, you will divide with 1000. Similarly, for liter to the meter cube conversion, we will also divide with 1000. So this will cancel, so these units become equal. But you have to take care of these things, because it is really important to take care of the units, because when you won't consider the unit, then definitely your answer will not come correct. So finally time will come around. Can anyone try to solve this by putting all the values, so that you also know whether it is whether the answer coming is correct or not. So anyone please try to solve this. Okay, I'll write it. It will become 0.5 of S naught into 5 plus 0.5 into 200 0.5 into 10 raised to the power minus 2 into 0.5 of S naught. This is 5. So when you solve this time will come around into 10 raised to the power 3 divided by 5. One more thing you have to take care here, that all the units here, because Vmax, the time given it is in second and we are using the Vmax. So here what the value of the time will come, it is in second, that need to be converted in hour, because they are asking about the 15 kg product per day. We'll also convert this in hour because the value must remain in the similar type of unit. So this value become very large in second, so when it will be converted into hours, it will become 5.83. So time is 5.83. This thing is really important here, that conversion of units. Okay, now time will come. Now what you have to find, you have to find the flow rate, and for flow rate you require the substrate concentration or the actual substrate. As you know that they have given x 0.5, so what it is representing, that only 50% conversion, so that's why x become 0.5. It means what they are asking for the substrate concentration 15 kg per day, that is the required condition. But because conversion is only 50% occurring, so actual concentration will become 15 divided by this factor 0.5. So it will become 30 kg per day, that the initial substrate required is 30 kg per day. But initially we have calculated the time in hour, so we also required this value in hours. So we know it will become 24. So anyone please let me know how it will come, what is the value of this. Okay, this will become actually 1.24 or 1.25. So this is the actual concentration that is required per hour. So how the flow rate will come? Flow rate is the actual concentration required and what we initially have. Because we must know like how it. Okay, I'll erase some part. So this actually required upon initial, that is the initial substrate concentration that is given, and now what it will come, this is the required 1.25 per hour, that is kg per hour. Initial what they have given is 200. Okay, let's write the unit kg per hour, and that 200 you can write is kg per meter cube, because I already told you that gram per liter is equivalent to kg per meter cube. Because we are converting both denominator and numerator, that's why this will become equal. So when you solve from here, this F will become 0.00625. And what is the unit of this? That kg will cancel out, meter cube per hour. This is the flow rate. Now let's go back to the question. What we are finding in this question? We have to calculate the volume. And how can we calculate the volume? Flow rate into time. Now we have both the things. First we have calculated the time by using this Michaelis-Menten equation and this CSTR equation, because this is only used in case of CSTR. When it will convert it into batch, then this time period will change and it will change the whole equation. Okay, so from there, what is the value of time we get? That is 5.83. Then how do we calculate the flow rate? From the substrate concentration what they have given and what is required to fulfill the condition. That initially they are asking for 15 kg per day, but how much conversion is occurring? Only 50%. So it means their actual required, it will become 30 kg. From here we calculated the F, that is initial concentration, sorry, actually required by the initial. This you can check from the formula also. So it will become around 0.00625. Now you have to put both the values in this equation and you will get the answer. That is, when you put the value of F, that is 0.00625 into time, what time it will come? It is 5.83. 5.83. You also have to take care the unit. So when you solve this, anyone please let me know what it will come. Okay, I'll solve this for you. Yeah, someone write it. 0.0364. Correct. But there is no option like this. So what to do? So you have to take care the unit here. So this value come in the form of meter cube. You can check it from here that it is given in R and this F value is meter cube. So meter cube by R into this value come in the form of meter cube. So when you convert this meter cube into liter, you have to multiply it by 10 raised to the power 3. So it will become 36.4 liter. So this is the final answer. I know this is a little bit lengthy question, but it is really simple. If you focus on the thing that you need to find out and what it is given, then you have to do all the things in sequence, then you will get the right answer. But everyone please try to focus this also, that this is given in a CSTR condition. You can also try to find out in the form of batch culture. So answer to this question is C. That is 36.44 liter is required to produce this 15 kg product per day. And this condition also important, that 50% of conversion is occurring. Suppose if they change the condition that only 60% conversion is occurring, here you can directly replace, you can equate S equal to S naught. But when this is given that 60%, then you have to find S equal to S naught into 1 minus X only. You can't write it equal to 0.5 of S naught. So you have to take care all these things while solving the question. So if anyone has any query, please let me know. Otherwise, we'll close this session.
Excuse me, ma'am.
Yeah.
Ma'am, I have a clarification from the first assignment.
Yes.
Um,
Hello?
yes, ma'am. In
Yes.
the 12th question,
Okay.
should I share my screen for the question, ma'am?
Okay, I'll check. Just let me know if anyone want to ask any question from the today's session. I'll tell you after 5 minutes.
Yes, ma'am.
Yeah, someone write something. Can you explain question number 8 again? Okay, let's go back. Question number 8. Okay, this is very simple question. What they have given. Satvika, you are asking about this question only, no?
Yes, ma'am.
Okay,
Ma'am, like, can you please explain it from that V1 divided by V2, that part, ma'am, from that part?
okay. From V1 divided by V2, either you have to do one thing, calculate the value of V1 and V2 separately, then divide it. Or if you want to, you don't want to do in the calculations, then you can directly divide. When you divide this, you can cancel out many things. That this, okay, I'll write it again. Sorry. This V1 value, that is 100 into 100 by 110, and here it will become like 2 into 100 into 100. You can write so that it will cancel out each other. Only to simplify the calculation, we cut this. It will become 105. So when you divide this v1 by v2, this whole part will cancel out each other, and this, it will become upon 1 by 110 into divided, because it is in denominator, so it will come in the denominator of this one, and this 105 will become in the numerator. So we have at the end 105 divided by 1010 into 2. So for the sake of this calculation, we have considered that this value come almost equal to 1. Sorry, here 2 is, and it's not necessary that you will consider it as 1. You can do one thing, you can solve this also if you want, but doing this will not help you in this, because they are only asking about how much times it will be coming, so you only need the ratio. So if we consider as a rough value that it will come around, suppose it will be coming around 9 1, so we are considering at 1, and this will become 1 by 2 is remaining here. So this 1 by 2 will give you the value of 0 0.5, that is v1 by v2 is equal to 0 0.5. It is not exactly 0 0.5, but because in this answer also or options they have given that is approximately symbol, that it is around 0 0.5 times of v2. So it is not like the exact value it will come 0 0.5. If you want to solve, it will come maybe around 0.47 or 49. So we consider it nearby equal to 0 0.5. Okay, anyone
Yeah,
have
okay, ma'am.
any other query in this assignment? Okay, if there is no question, let's close today's session.